All right. Hello, I am Ria Talwar and I'm presenting from Seattle, Washington. My presentation is about explaining genetic variation in the Pacific white-sided dolphin through environmental factors. I chose to study this topic because I'm very interested in genetics and how an organism's genome can be impacted by its environment. I think that this topic has become especially relevant as the natural habitats of many organisms are rapidly changing and deteriorating due to climate change. So, environmental changes driven by pollution and climate change have affected many species globally. The Pacific white-sided dolphin is particularly threatened by water pollution caused by toxins and debris in the water. These problems are the product of human actions in areas along the coast. To begin my research, I looked at environmental threats that the Pacific white-sided dolphin faces and discovered that pollution and temperature variation were notable dangers to the species. Some populations in the Gulf of California have already been reduced due to warming waters in the area, or as the species prefers cooler waters. Microplastics can also be dangerous for the species, as they are for many ocean-dwelling organisms. Strong environmental pressures like this can impact the genetic structure of species in affected areas. Examining these genetic differences can provide insight into how organisms adapt to these changes to survive. While researchers have studied genetic differences between populations of the Pacific white-sided dolphin in Japan, they failed to explore the potential environmental drivers of these differences. Previous research using the same data found that two distinct genetic clusters exist, but they did not identify the causes behind the distinction. When beginning my project, I did extensive research on past studies about genotype-environment correlation to see if any relationship had been found in previous studies. I found that a correlation had, in fact, been found in multiple organisms. For example, certain species of corals in the Great Barrier Reef were found to have different genotypes depending on their environment. There was a clear relationship between coral genetics and the water clarity or temperature of their environment. Similar results occurred in species of fish, indicating that my investigation was likely to find some correlation. My research aims to identify the major environmental factors impacting the genetics of the Pacific white-sided dolphin. Due to the notable presence of microplastics in the ocean surrounding Japan and the variation between densely populated and isolated areas, I initially hypothesized that this would have the most notable impact on the genetics of the Pacific white-sided dolphin. This research will increase our understanding of how the species has responded to the increase in water pollution and other environmental changes that occur due to human actions. To begin my research, I found an existing study of the Pacific white-sided dolphin that sequenced the genomes of dozens of organisms from across Japan. This study identified two genetic clusters but had not investigated the cause behind the clusters. Using this sequencing data, I created a genetic distance matrix that could be analyzed alongside environmental data gathered from the locations of each population from the study. Using the sites given for each organism, I collected environmental data for salinity, surface temperature, microplastic levels, and current to analyze alongside the genetic data. Using R, I generated a heat map, several principal coordinate analyses, and a distance-based redundancy analysis to visualize the relationship between different environmental variables and the genetic distance between organisms. In the heat map shown on the slide, we can see that there is more substantial similarity between the organisms on the upper left because the blue coloration indicates lesser genetic distance. The blue stripe down the middle indicates no genetic difference because that is where organisms are being compared to themselves. Darker red values show greater genetic distance. After generating a heat map, I created several principal coordinate analyses, or PCAs, to look at the correlation between environmental variables and genetic distance. PCAs visualize the similarity between samples in multidimensional space. If 2 points, each of which represents a dolphin, are closer together, they are more genetically similar. The graph can then be colored on a gradient by an environmental variable, and if the colors appear to be in a specific pattern, it suggests a relationship between genetics and that variable. The PCOA shown on the slide is colored by genetic cluster, which is a binary category determined by the authors of the original study. There's a clear division between the light and dark points, which matches the expected correlation because genetic cluster is based on genetic distance. This PCOA shows the relationship between current stability and genetic distance. While the relationship is not as clear as it was for genetic cluster, there is a pattern shown by the graph. Darker points with lower current stability values are shown more in the upper quadrants of the graph, while lighter points with higher current stability values are clustered in the bottom quadrants. This figure indicates a correlation between current stability and genetic distance. This final PCOA is colored based on salinity values and shows a less defined distinction than in the previous two figures. While there are some groupings of lower salinity values or darker points in the bottom quadrants of the figure, the pattern is not so distinct that a correlation is immediately obvious. To take a deeper look into the relationship between microplastics, several other variables, and genetics, I created a DBRDA, or distance-based redundancy analysis. This figure is generated from the genetic distance matrix and environmental data, which is standardized to avoid bias when comparing different variables. An RDA shows how genetically different samples are for specified variables, in this case, environmental variables. This provides a way to see if certain variables can explain some genetic differences. The length of the arrow indicates how strong the correlation is, and the direction indicates where the greater values are. The graph indicates a strong correlation between current and genetics compared to other variables, and salinity also shows a notable correlation. Among the remaining variables, there are varying amounts of correlation, with surface temperature having one of the weakest correlations. After obtaining my initial results from the RDA, I set up a random forest, a machine learning model, to help confirm the outcome. Using the random forest package in R, I divided the data into training and testing sets, with 75% of the data in a training set and the remaining 25% in the testing set. The training set was used to train the model to determine the genetic cluster of an organism based on given environmental data. The testing set was then used to test the model by having it predict known genetic clusters based on the environmental data provided, which it achieved with about 79% accuracy, which is reasonably high. Once I completed the training and testing the model, I was able to extract the variable importance to determine which environmental variable had the most notable impact on the predictions made. When I examined the variable importance established for the random forest model, as visualized in the figure on the slide, the results showed that current stability was the most decisive variable, and salinity also had a notable impact. To further understand the results of the DBRDA and random forest, I looked at the data distribution for current stability and salinity. The two box plots on the screen show the data range and distribution for current stability and salinity. Both box plots show a distinct data range between genetic clusters. This can help explain why these two variables are major factors in determining genetic cluster. However, we need to understand why these variables impact genetic variation and how they affect the Pacific white-sided dolphin. So from my initial results, it appears that current surprisingly has a significant correlation with the genetics of the Pacific white-sided dolphin. Polybrominated diphenyl ethers, a type of microplastic, also have some impact, but it differs from the other type of microplastics, polychlorinated biphenyls, which has a weak connection to genetics. Temperature has a less notable impact than was initially hypothesized, potentially due to less variation among the region that I looked at, but salinity does have a sizable impact. These results were confirmed by the random forest model, which showed a similar variable importance to the RDA. The next steps to understanding these results are to research why current may have such a strong impact on genetics and determine the significance of these results. Current impacts distribution of heat and nutrients, along with affecting migration patterns and organisms present in some areas. This could affect the prey that's available to dolphins in certain areas. Ocean current impacts a variety of environmental factors, particularly the organism and nutrient diversity, which likely leads to its genetic impact on the Pacific white-sided dolphin. Further research will help provide insight into the causes behind these connections or lack thereof for different variables. Thank you all for listening.
Thank you very much, Ria. So, quick question. Are the results that you discovered enough to affect policy and rulemaking, or, as you suggested, is more research needed before this can really have an impact on the environment and choices that we make that can impact the dolphin?
Yeah, well, I definitely think it's a good idea to have governments look at these results to make decisions on policy, but this study is only focused on a certain area in Japan, so it like right now cannot be applied to a wider scale. You would need to investigate populations of dolphins all across the world instead of focusing on a specific region.
Perfect. Thank you so much.